I'd like to start by briefly introducing DEFI USA and our mission. DEFI stands for Docteur Français à l'International, which means Global French PhDs. And the word défi in French means challenge. DEFI is a network that brings together scientists who have obtained their doctorate in France and are pursuing their careers in the US, and also includes PhDs who are back in France and who wish to share their past experiences in the US. Our mission is to help PhDs newly arrived in the US to feel welcomed and included, to share information on professional opportunities for PhDs in the US and also in France, and to share ideas, advice, and personal experience of living in the US. To learn more about DEFI or join our network, please visit our website. My DEFI colleagues, Audrey Boyer and Laurie Hervieux, are now taking care of the chat, and they will share the link with you in a moment. This event, Cross Science, is organized by DEFI in partnership with the Office for Science and Technology at the Embassy of France in the US, and showcasing scientists who hold a PhD from French universities. Our three experts today will present their work in the field of diabetes, memory, and water resources. So before diving into the science topics, I'll share quickly about the structure of today's conference. Our three speakers will give their presentation, and then we will have a question and answer period at the end of the three talks. So feel free to type in your questions in the chat anytime, and please let us know which speaker the questions are for. We will collect all the questions throughout the event, and I will ask them at the end during the discussion. So without further ado, I would like to welcome our first speaker, Dr. Eric Lonchi-Yimagu. Dr. Lonchi is a postdoctoral fellow at the Global Diabetes Institute at Albert Einstein College of Medicine in New York, supported by a fellowship grant from the American Diabetes Association and enrolled in Einstein's clinical research training program. For the past 10 years, he has been actively involved in projects related to the epidemiology and pathophysiology of diabetes, both at national and international levels. Dr. Lonchi received his master's degree in nutritional biochemistry for the University of Yaoundé in Cameroon, and his PhD in physiology and pathophysiology from the Université Pierre et Marie Curie in Paris, France.
Okay, so thank you so much, Pauline, for this nice introduction. I really appreciate. And I would like to thank everyone here for joining this meeting. Indeed, it is a pleasure and honor for me to give this presentation. And I hope each of you had a good breakfast, a good lunch with enough of vitamin D, because as you can see in the background of my slide, our world is being ravaged by a tsunami of diabetes. So today I'm going to talk about the role of vitamin D in the prevention of this global diabetes epidemic. But before I go into that, I would like to share a funny slide with you. This is a picture of me. Actually, I developed an interest for science very early in my life. I was born in a small village called Bacham in the western region of Cameroon. Cameroon, it is in Central Africa. So ever since I was a kid, I love health, I love medicine, I love science fiction. My dad is a doctor, and watching him work with diabetes patients as a kid inspired me. It still does today. And this is why I'm so excited, I'm so happy to celebrate La Fête de la Science with each of you today. So I did my master in biochemistry in Cameroon, studying the link between diabetes and nutrition. And at that time in Cameroon, it was popular to believe that eating quail eggs could cure diabetes. How many of you know quail eggs? Well, it was popular to believe that quail eggs could cure diabetes. And even my uncle was drinking these eggs, and this was really expensive. So I was curious to know if this is true or not. So I performed a research project to test it. My paper was published showing that quail eggs did not cure diabetes at all. I was really sad. You know why? Because I missed the Nobel Prize. After my paper was published, the consumption of quail eggs dropped significantly in Cameroon. And I am sure those who are in the business of quail eggs are still mad at me today. But that was not the goal, right? The goal for me was to do something that could help my uncle and millions of people suffering from diabetes in the world. Because here's the problem. There is a tsunami of diabetes in the world. If the current trend continues, if nothing is done, in 25 years from now, 700 million adults in the world will have diabetes. And as you can see in this map, no country, no continent is immune from diabetes. Whether you are rich or you are poor, you are affected by diabetes. Let's take a couple of examples. In North American and Caribbean, where we are now, the number of adults with diabetes is expected to increase by 33%. In Africa, where I'm originally, by 143%. In Southeast Asia, where I've been traveling for the past 4 years to study nutrition and diabetes, it is expected to increase by 74%. In Europe, where I did my PhD, by 15%. And every second, one person died from diabetes in the world. So how many of us want to be part of these 700 million of people that will have diabetes by 2045? No one, right? So there's a need to support science. There is a need to support scientists. There's an urgent need to better understand how diabetes progresses and to develop solutions for the prevention. And I'm one of those who believe that we are what we eat, and nutrition could play a role. So I hope my presentation will do three things adequately well. What is vitamin D? What is type 2 diabetes? Could vitamin D help to reduce the risk of type 2 diabetes? Well, vitamin D is a group of fat-soluble responsible for increasing intestinal absorption of calcium, magnesium, and phosphate, and many other biological effects. The most important sources of vitamin D is endogenous production in the skin when we are exposed to UV radiation. But you can also find vitamin D in many different foods like fish, milk, cheese, beans, or also from supplement. But intriguingly, it is estimated that about 1 billion of people worldwide have vitamin D deficiency or insufficiency. Now, what is vitamin D deficiency? Vitamin D deficiency is defined as a serum level less than 20 nanogram per milliliter, and vitamin D insufficiency is defined as serum vitamin D level between 20 and 30 nanogram per milliliter. Above 50, it is considered above the normal range. What is type 2 diabetes? There are different forms of diabetes, but type 2 diabetes represents the most common form. And type 2 diabetes just means that a person has too much glucose or sugar in the blood. And this is because the pancreas does not produce enough of insulin, or the body cannot use the insulin well enough. And this phenomenon is called insulin resistance. And when you are insulin resistant, you are more prone, you have more chance to develop type 2 diabetes. So now, does vitamin D repletion have beneficial metabolic effect? And if yes, what will be the potential mechanism? And I hope at the end of my presentation, I will be able to answer this important question. Here's our hypothesis. Adipose tissue expansion, this is fat. Adipose tissue expansion during obesity is associated with adipose tissue inflammation and fibrosis. And these two factors lead to insulin resistance, which is not good for the body. What we know is that vitamin D receptors are present on adipocytes, are present on fat. And these cells have the potential to produce biological response to vitamin D, including the reduction of inflammation. So what we propose is that vitamin D repletion in obese, insulin-resistant subjects with vitamin D deficiency will reduce fat inflammation, fat fibrosis, and altogether, this will lead to an improvement of insulin resistance. So our goal was to assess the effects of vitamin D repletion on adipose tissue inflammation. This is adipose tissue inflammation, adipose tissue fibrosis, and on systemic insulin resistance. So we did a study in human and in rodent. So it is based on our human study that we develop more study in rodent in order to explain the potential mechanism. So now for the human study, we include 19 obese subjects with vitamin D deficiency. Remember, vitamin D deficiency is below 20, and these subjects were insulin resistant. These 19 subjects were randomized. They were divided in two groups, the vitamin D treatment group and then the placebo group. So what we did is that we replaced vitamin D in the vitamin D treatment group at two different levels. So we have 3 visits in total. The first baseline visit where each subject arrived with obesity, with vitamin D deficiency, and with insulin resistance. The second visit, that was after 3 months, where we replaced vitamin D from low level to the normal range. This is the second visit. And then during the third visit, this was achieved after 6 months in total, we replaced vitamin D from the normal range to above the normal range. But in the placebo group, they received placebo and then they remained vitamin D deficient during the 6-month study. This is a figure just to show you the representation of our study design. Here, the treatment group, and here the placebo group. We have 3 visits. First visit in blue, second visit in orange, and then the third visit in gray. So in the treatment group, there were vitamin D deficient, as you can see here in the blue, at the first visit. So we replaced vitamin D to the normal range. And this was achieved after approximately 3 months with weekly administration of vitamin D at the dose of 40,000 international units orally per week. And then vitamin D repletion above the normal range from the blue to the gray was achieved after 6 months. As you can see here in the placebo, because they were receiving placebo, they started with low vitamin D. They remained vitamin D deficient during the 6-month study period. And then our goal was to see whether vitamin D repletion here could improve, could reduce fat inflammation and fat fibrosis that I described before. That is why during each study visit, for each subject, we collected fat biopsy in the abdomen. We collected fat biopsy to assess adipose tissue metabolism. During each study visit, we assess insulin sensitivity using the glucose clamp techniques. So we did that both in the treatment group and then in the placebo group. This is just a description, a simplified description of the technique that we are using. This is the best ever technique in the world to assess insulin sensitivity in humans. And with these techniques, we are able to determine what is insulin sensitivity at the level of the liver and what is insulin sensitivity at the level of adipose tissue and muscle. So we have 2 main endpoint measurements: hepatic insulin sensitivity, liver, and peripheral insulin sensitivity adipose tissue, and muscle. Just like I said before, for each subject, during each study visit, first, second, and third visit, both in the treatment group and then in the placebo group, we collected fat biopsy during each study visit, just to see whether in the treatment group, we could reduce fat inflammation, we could reduce fat fibrosis. We are not good for the body, because when you have more fat inflammation, more fat fibrosis, you are more prone to develop insulin resistance. And when you develop insulin resistance, you are more prone to develop type 2 diabetes. Now, what are our results in the human study? So in the human study, we found that vitamin D repletion improve liver insulin sensitivity in obese, insulin resistant, and vitamin D deficient human, which is great. We found that vitamin D repletion reduce fat inflammation, fat fibrosis in obese, insulin resistant, and vitamin D deficiency. What happened in the placebo group? Remember that in the placebo group, they remain insulin resistant, they remain vitamin D deficient during the 6-month period. And we realized that during the 6-month period, there was a trend toward increasing on their insulin resistance. There was a trend toward increasing on their fat inflammation, on their fat fibrosis, which is not good. And this suggests that vitamin D deficiency over a long period of the time could increase insulin resistance, could increase fat inflammation and fibrosis, and could lead to the development of type 2 diabetes. So now, in order to understand the mechanism by which vitamin D could improve insulin sensitivity or adipose tissue inflammation, we did some complementary study in rodent. And this is the beauty of translational research. And what we did is that we have 2 groups of mice. In one group, we remove vitamin D receptor in adipocyte. Remember, in the first couple of slides, I said that vitamin D receptor are present on adipocyte. And what we did in these mice is we remove vitamin D receptor, and then we compare these mice with the normal wild type mice, just to see what is happening in this group of mice. Now, we also perform the glucose clamp technique, which is the gold standard for the evaluation of insulin sensitivity. And what we realize is that compared to the wild type mice, the adipocyte-specific vitamin D receptor knockout mouse model developed here an increase. There was an increasing on liver insulin resistance. There was an increasing on adipose tissue inflammation and fibrosis, and this clearly matched the result that we found in human. This is just to say that if we were able to remove vitamin D receptor in human, in adipose tissue, this human will develop insulin resistance, they will develop adipose tissue inflammation and fibrosis. But because we cannot do that in human, this is why we move into mouse, and these findings match the result that we observe in human. So this complementary human and rodent study support a beneficial role of vitamin D repletion for improving liver insulin resistance and reducing fat inflammation and fibrosis in targeted individuals. To answer to the original question that I asked before, does normalizing vitamin D level could have metabolic benefit in target individuals? So with the current global obesity epidemic putting millions of people at risk of type 2 diabetes, correcting vitamin D deficiency in these at-risk individuals could have a major public health impact. What is the take-home message of this study? Those who are obese, insulin resistance, and vitamin D deficient may simply take vitamin D supplement. This is a cheap, safe, and effective solution that could make a real impact on the prevention of diabetes. For those who want to have more information about this study, our paper was recently published in the Journal of Molecular Metabolism, which is great. And I would like to thank our team at the Global Diabetes Institute and the funding sources of this study, especially American Diabetes Association, who continue to support my research. Thank you so much for your attention.
Thank you so much. Thank you. That was a really nice presentation and very clear. I learned a lot. Thank you very much. So if you still have questions, please continue putting them into the chat box and we'll discuss them at the end. Now I'd like to hand over the mic to our second speaker, Dr. Chloé Bouarab. Dr. Bouarab is a postdoctoral fellow at George Washington University in Washington, D.C., working on sex differences in stress-related pathologies. Dr. Bouarab received her master's degree in neurobiology and neuroscience from Université Paris-Descartes and her PhD in neuroscience at the Université de Bordeaux in France, working on the molecular mechanisms underlying the switch from normal to pathological fear memory. Thank you.
So good afternoon, everyone, or good evening. Thank you, Pauline, for the presentation. And yes, just as you said, today I'm going to talk to you about how your emotions can modulate your memory. And so in a really simple way, memory is the ability for an organism to encode, retain, and recall past experiences. And there are different types of memory. For example, remembering something from our past, it's not the same way, same thing as remembering how to ride a bicycle. So we're going to talk about declarative memory in one situation and about non-declarative memory in the other one. And all those types of memory rely on different brain regions. So today, I'm going to focus on declarative memory. That includes semantic memory, or all the historic facts that you can learn in school, etc., and episodic memory, that is like your own personal memories. And this type of memory, the declarative memory, relies on a brain region that we call the hippocampus. And so the same way it exists different types of memory, all your memories are not encoded with the same strand. And it's really easy to picture. Like if I ask you what you had for lunch, like Monday 2 months ago, no one will be able to respond. But if I ask you where you were when you heard about the terrorist attack against Charlie Hebdo or the 9/11, the memory will kind of pop up in your mind right away. And the same way, extremely happy events, such as a wedding or the birth of your kids, they are deeply rooted in your memory. And scientists call this phenomenon flashbulb memory. And if we plot that on a graph, you can see here that indeed, when you increase the stress level or the emotional intensity, you improve the memory score. And that's what we call emotional memory. And so studies in humans and in animals have shown that there are two key brain regions involved in this emotional memory: the hippocampus that you can see here in green and the amygdala in blue. And so scientists have shown that these two brain regions can influence each other. So in the first study, people were presented with cards representing different shapes and colors. And people are warned that every time they're going to see the blue square card, they will receive a mild electrical shock on the wrist. So while we present the cards to the people, we record their fear reactions at the same time. And even if people actually never receive a shock, you can see here that every time they see the blue square card, they express a fear reaction. And so the hippocampus is necessary to the acquisition of a declarative memory of this event. What I mean is the hippocampus is necessary to be able to say, I'm afraid, every time you see the blue square card. But the fear reaction that we measure depends on the amygdala. Like, data have shown that a strong fear reaction is associated to an increase of the activity in the amygdala. And so to know a bit more about this brain region, another study focused on the memory score of a patient who had a calcification of the amygdala. So this person has a non-functional amygdala. And so in this study, subjects watched 2 video sessions, one with neutral clips, like travel story, and another one with negative emotional content, like violent crimes. 3 weeks later, subjects are contacting over the phone and being asked to remember as many clips as they can. And so as you can see on this graph, controls remember more emotional clips than neutral ones. So emotions did improve the memory of the control patient. But when the amygdala is not functional, as you can see here, you lose this difference. So we know now that amygdala is important in strengthening the emotional memory, and that there is a dialogue between the hippocampus and the amygdala, and that this dialogue is crucial for emotional memory. So what we learned so far is that stress can promote memory. And from an evolutionary perspective, it's completely adaptive. And specifically when this emotional memory applies to a dangerous situation. And it makes sense if you think about the time when people were hunting, for example. If during a hunt you survive to an encounter with a saber-toothed tiger, it's critical that you remember the event to make sure that you will know what to do the next time it happens. But sometimes after an emotional event extremely intense, such as a war or a rape, some individuals will switch to a pathological form of emotional memory. And so post-traumatic stress disorder, or PTSD, is one example. So PTSD patients display, among others, 2 main characteristics. An amnesia for what we call the peritraumatic contextual cues that I will explain in a moment, and hypermnesia for a simple salient element. So what does that mean? The amnesia refers to a deficit of contextualization. Patients are usually unable to construct like a linear memory of the traumatic event. And this lack of contextualization leads to an inability to restrict fear to the traumatic context. As an example, you can think about a soldier coming back to the civil world and afraid of the noise of fireworks because it reminds him gun shooting. And so because of that, patients display intense and uncontrollable fear reactions in neutral context. And so once again, being afraid of during a firework is not a normal physiological response. And so all this leads to flashback and nightmares where patients will re-experience the traumatic event again and again. And so we talk about hypermnesia because often the emotional memory has focused on a simple salient element which was present during the trauma, but not predictive of the trauma. So just like after being attacked by someone with blue eyes, you shouldn't be afraid of all blue-eyed people, to put it in a really simple way. So if you remember from the previous slides, we established that the hippocampus was important for declarative memory, being able to form like a conscious memory of an event. And the amygdala is involved in emotional memory. And so our hypothesis was that during the formation of a normal fear memory, and I say normal because it's normal to be afraid of a threatening situation, you have kind of a balance between those 2 brain regions. But when you switch to a PTSD-like memory, the involvement of the hippocampus is kind of reduced, and the activity in the amygdala gains a lot of weight, creating like a dysregulation in the system. So in order to test our hypothesis, we need an animal model of this PTSD-like memory. So on day 1, mice are exposed to a new context, the first one, just for them to habituate and not being afraid of it anymore. The next day, it's the fear acquisition. So mice will receive 2 mild food shock, electrical food shock, and between those two food shocks, they will hear a tone twice. But, and it's really important for the next part, the tone is never associated with the food shock, so the animal shouldn't be afraid of it. And half of the animals will receive right after the fear acquisition a second stress, just to mimic a traumatic event. Then on day 3, we will proceed to memory test. So first, animals are re-exposed to the tone in the neutral context of day 1. Then 2 hours later, they are re-exposed to the traumatic context. And we measure their fear reaction in each of these contexts. So now, how can you tell a mouse is afraid? Well, what we do is we measure freezing. And freezing is a behavioral response where the mouse is completely immobile except for the respiratory movement. So when we do that, a mouse showing a normal fear memory should not display fear to the tone because it's not predictive of the shock. But the mouse should be afraid of the context where it received the shock. On the opposite, a PTSD-like mouse will be afraid of the tone, just think about it as like its own firework, but will be less afraid of the context. And that's exactly what we see on this graph. So a normal fear memory, you have no fear to the tone, and you have a high percentage of freezing, a high fear reaction to the context. Whereas in red, you have the PTSD-like map, the one submitted to the traumatic event, and you see that you have an abnormal fear response to the tone associated to a reduced fear response to the context. So now that we have a good animal model, we can start to wonder what happens in the brain that can explain this abnormal memory. So here on the right, you have a mouse brain. And if you slice it, you can have access to the different brain areas. So today I'm going to focus on the hippocampus and the amygdala. And when you analyze neuronal modification, you can focus on different parts of the neurons. You can focus on the soma area, the number of nodes or number of branches, the length of these branches, of the termination. So for today, I'm going to talk exclusively on the number of nodes, the number of branches. So first, let's just take a look at the hippocampus. So if you remember, it's the brain area involved in declarative memory. And so hippocampus is important to formulate a conscious memory. So in black here, you have animals with a normal fear memory. And in red, you have the PTSD-like ones. And so as you can see, the switch from a normal to a PTSD-like memory is associated with a decrease in the complexity of the neurons. Neurons have less branches. And so what it means is that these neurons will be less complex and less likely to establish connections with other neurons. And so the overall communication will be altered. And so now if we look at the amygdala, we see the exact opposite pattern with an increase of the neuronal complexity. So our hypothesis was correct. Like the switch from a normal fear memory to a PTSD-like memory is associated with a lighter involvement of the hippocampus and an overwhelming amygdala. But now what we want to know is, okay, can we kind of prevent the development of a PTSD-like memory after exposure to a traumatic event and still develop a normal fear memory? So I showed you that between a normal fear memory and a pathological one, we have like a loss of neuronal complexity in the hippocampus. And so our idea was, okay, maybe if we use like a pharmacological compound to block this loss of complexity, can we prevent the development of a PTSD-like memory? So to answer this question, we use the same protocol as before. You have the fear acquisition with half of the animals submitted to an extra stress, and you have the memory test the next day. But this time, 30 minutes before the fear acquisition, animals receive directly into the hippocampus a drug that will block the loss of neuronal complexity, or a vehicle just as a control. And so here are our like regular groups. So in white, you have the normal fear memory with no fear response to the tone and a high fear response to the context. Sorry about that. And in red, you have the PTSD-like mouse with an abnormal fear response to the tone and a reduced fear response to the context. But what is really interesting is our last group, the one with the dashed lines. So here, it's a group where the animals have been exposed to a traumatic event, so like food shock plus the second stress. But here, as you can see, blockade the neuronal loss allowed us to maintain a normal fear memory with animals looking just exactly like our controls. So yes, we can prevent the development of an abnormal memory after a traumatic event by maintaining a normal activity in the hippocampus. And so altogether, this data show that like a kind of equilibrium between the hippocampus and the amygdala is really important for normal emotional memory. And so finally, I would like to thank the DEFI Association for the opportunity to share this work with you today, and thank you all for your attention.
Thank you very much. That was extremely interesting. And I feel like a topic like that following Veterans Day is on point. So thank you. So now I'd like to hand over the mic to our final speaker, Dr. Fadji Zaouna Maina. Dr. Maina is an associate Earth scientist at NASA in the DC area. And before joining NASA, Dr. Maina worked on the development of mathematical models to address water resources issues in California, France, and Italy, using high-performance computing and satellite data to understand the impact of climate extremes and wildfires on water resources. Dr. Maina was included in the Forbes magazine's 30 Under 30 list in 2020 and was qualified as a rising star in civil engineering by the MIT. She received her master's degree in environmental engineering and her PhD in hydrology from the University of Strasbourg in France. So for this presentation, I have a video to share with you.
Water. Up to 60% of our body is made of water. More than 70% of our Earth's surface is covered with water. Water is essential to our daily lives. We need to prove it. It used to be accessible for everyone, clean and safe. But today, with the onset of climate change, water is threatened in many countries around the world. Pollution in Asia, water scarcity in arid regions, flood in many countries. The consequences of climate change around the world are affecting our water resources and changing the water cycle. It becomes critical to tackle these issues before we run out of water, and you will all guess what will happen next. We know the water cycle since the dawn of our civilization, yet it's incredibly hard to reproduce it and to fully understand the interaction between the different components of this water cycle. We need to be able to reproduce it to better understand how perturbations are going to affect it and how we should better prepare ourselves to address these situations. To understand the water cycle, we use mathematical models, a set of equations that describe every movement of water from the moment the water left the atmosphere to reach the ground as a rain or snow, when this rain is intercepted by vegetation, or when this rain reaches the soil to recharge groundwater or to increase river stages. We make sure that our mathematical models are able to reproduce what's happening in reality by combining them with measurements performed on Earth, such as groundwater and river stages, but also in space by satellites. Today, Many satellites measure different components of the water cycle at an unprecedented resolution and in near real time. We create a virtual water cycle, and we can control everything that's happening inside and see how this is happening. We use this high fidelity, high resolution model to capture the movement of water on Earth and to understand how perturbations such as wildfires are affecting the water cycle. Wildfires are becoming increasingly frequent and devastating. You all remember wildfires in California, wildfires in Australia. Unfortunately, these wildfires are projected to increase with climate change. We have no other choice than learning how to deal with wildfires. Wildfires damage vegetation. After wildfires, the forest disappears, and we go all the way from forest to birds with no trees. We use our model to reproduce these changes, and we are able to demonstrate that. Because wildfire is damaging vegetation, wildfire is going to increase groundwater and river stages. Without vegetation, in the absence of canopy, no precipitation is going to be intercepted. Rather, that precipitation is going to reach the soil, to recharge groundwater and river stages. But who says water abundance says also disasters, such as floods. So we may hold ourselves for stating that every cloud has a silver lining. This is a glimpse of what our model can do with the use of satellites. We can use this model to project how water is going to look like by the end of the century at an unprecedented resolution and high resolution in time. Nonetheless, the future is complex and will bring situations we have never seen before, which may be out of the scope of our current knowledge. That's why we are deploying a lot of effort to improve the accuracy and the precision of this model to be ready for the future, to build a sustainable environment where everyone will have a safe and clean water, not only for us, but for the next generation. That generation deserves a better world. Let's clean our mess. Thank you very much for your attention.
Thank you very much for this wonderful video. It's true that when we think about NASA, we think about space and we don't think about how satellite data can be used for things that are way beyond space issues. So thank you very much for this really interesting presentation. So now I'm opening the floor for questions. So feel free to continue typing questions in the chat. And I guess I'll start with a question to Dr. Lonti. Do you have? I have a question from the chat. Do you have an idea of what is the respective part of type 2 versus type 1 diabetes in the diabetes pandemic?
Yeah, first of all, I feel like we have a lot of experts from doing diabetes research here because there's a lot of questions that I already saw. Yeah, so in terms of prevalence, type 2 diabetes represent approximately 90% on other form of diabetes, and type 1 diabetes is 10%. But there are some form of diabetes, including gestational diabetes, ketosis-prone diabetes, which is a rare form that is commonly found in population originally from Africa. There's malnutrition-modulated diabetes that is commonly found in low-income country like in India. So, but type 2 diabetes represent 90%, and type 1 diabetes represent like 10%, yeah, of the global diabetes epidemic.
Thank you. I have a question from the crowd to Dr. Bouarab. You showed results from a fear reaction test. Can you tell us about the way you measure the activity of the different parts of the brain during that test? You're muted.
Better? The one with humans or the one with animals?
If you could go over both, that would be great.
So in humans, that are not my data. I never work with humans. But basically, you just use fMRI. So you have, I think I have maybe a slide now. You have, it's just like a MRI, but what people do, you're going to present to the patient different images or different sound, and at the same time, you have, you can have access to the brain activity. And people do that with how much glucose each brain region consumes. And so that's the way you can tell which brain region is more active during the task. So it's for humans. For animals, what I presented, unfortunately, the animals were sent to a better world because I needed the brain. But what you can do is just implant some electrodes in the brain, and the mouse is still alive, the mouse is completely fine. And while the mouse is behaving, you can record the cellular activity at the same time. So just depending on where you put your electrode, you can record the brain activity.
Thank you. I have another question for Dr. Maina. The question is, which satellite do you use to observe wildfires?
So we are using a bunch of satellites because we are simulating the water cycle. But to really understand like where the fire happened, we basically use a database coming from the California Department of Fire, and then these are using just cartography to be able to do it. Then there is satellite data such as MODIS, and MODIS is monitoring like changes in vegetation, and you can see from there like how the vegetation has changed. And then from there, we can assume how the vegetation changed based on the fire condition.
Thank you. Then I have another question for Dr. Langi. We had questions about quail eggs. Do the quail eggs contain a high concentration of vitamin D?
Well, for this study, actually, we had 4 groups. We had one group that were diabetes and we were receiving quail eggs. We had one group that were diabetes and were receiving chicken eggs. So we compare the effect of quail egg supplementation or chicken egg supplementation, and we do realize that those who were taking quail eggs had higher bad cholesterol compared to those who were taking chicken eggs, but we didn't measure the concentration of vitamin D in quail eggs or in chicken eggs. But at inclusion, those chicken or those quail eggs were coming from the same farm. So we didn't measure the level of vitamin D in quail eggs. Yeah.
Okay, thank you. Knowing that there is a link between vitamin D and sun exposure, could a vitamin D or could sun exposure be prescribed to people who have a risk for type 2 diabetes?
Well, I won't say that because there's a lot of controversy regarding what could be the effect of sunshine, because some people say that when you expose yourself to too much UV from sunshine, you have more risk to develop cancer. That is why it is more people propose to take, instead of going to sunshine, to take supplement like from vitamin D supplement or from food. Yeah, instead of UV, because UV you have more risk to develop some form of cancer.
Yeah, that makes sense. Thank you. Thank you very much. I have another question for Dr. Bouarab. We had a question related to false memories. You explained the difference between normal and pathological memory, and now the question is more how does hippocampus versus amygdala paradigm is working in relation with false memory. So when people recollect things but not accurately. So I wanted to know what you were like thinking about that, and if that can relate to what you are studying.
We did some experiment like this during my PhD, and there are two other brain regions that we call the lateral septum and the medial septum that are really involved also in like this general loop. And those are extremely important for those like false memory, just because the hippocampus can be seen as like important for contextual memory. But you're gonna have like the amygdala and in some extent the lateral and medial septum can be important for memory, but with like more simple elements. And so sometimes when you, depending of which brain regions you will put forward, let's say, with like a gain more, weight more important, you can modify the memory. And it was in mice, so it's not the same as in humans, but you can kind of play around with the memory, just playing with which brain region will be more activated than the other ones. It's definitely possible. Like I remember an experiment where so the mice were, it was kind of the same, like we're put in the in a context with a tone, nothing to do with the shock. And just by increasing the activity in the lateral septum, you could the same way kind of modify the memory that the mouse had from the event. And instead of being afraid of the context, the mouse would be afraid of the specific tone. So just by modifying the activity in different brain regions involved in this like huge loop important in memory, you can modify the memory itself.
That's fascinating and a bit terrifying too.
And a bit scary, I agree.
Yes, thank you very much. I have another question for Dr. Maina. Can you already see damages or changes in the water cycle after those very recent and huge wildfires that happened in California and Australia?
Yeah, actually, we can see it, especially like changes from wildfires, because like how the way that wildfire changed, like the vegetation and the landscape, it's really it's a lot. So just right after the wildfire, usually wildfire happen like this is a wildfire season basically in California and in summer, and then starting from like fall, we will start to have some rain, and then like just after like the first rain, we have flood coming in California, which is like quite common, and then landslide, and all these are like something that we are observing right now, and we show it with our models. And then we know that also we see like how the extremes also how they are changing in California, because we know that like sometimes, I'm talking about California because we basically focus ourselves in the United States and California. And because we know that like some years are very dry in California, no water, and then it becomes suddenly like very wet. So all this kind of aspect of climate change, we are observing this now, and we know the consequences. The point is like, we know that they are going to increase in the future. So we are trying to handle this increase in the future.
Okay, thank you. Thank you very much. I have one final question for Dr. Lanty. So in the study that you showed on your slides, you were showing patients receiving supplementation and the question was, do you see an increase in vitamin D in adipose tissue in those patients receiving the supplementation?
Very good question. So actually, what we did is we measure vitamin D receptor within the adipose tissue. And what we found is that in the vitamin D group, when we supplement vitamin D level from low to the normal range, there was that upregulation of vitamin D receptor. So to answer to this question, yes, there was an upregulation on vitamin D receptor, specifically from the first to the second visit and from the first to the third visit. Yeah. But in the placebo group, there was not effect. So vitamin D levels remain similar in the plasma, and the vitamin D receptor within the adipose tissue was not upregulated, just like in the vitamin D group.
Thank you. I have one final question for Dr. Bouarab. One funny question that you can answer if you want to. Should we scare people during conference presentations in order to make sure attendees will better remember the results? Is that your technique?
I should start my presentation with like a scary image or something, maybe like just to get people attention. It might work.
And if I can add one supplemental question, do you know of any treatments or therapies that are currently available for treating PTSD symptoms?
Yes, so treatments, like medical treatments, don't work really well right now. Most of the time, PTSD patients receive like antidepressants, and it doesn't work for a lot of people. What seems to work is, and it's related to what I showed, like the hippocampus seems to be really important because you don't want to erase a memory about an event like this because it's part of who you are. So, but instead of having the memory being like nightmares or flashback, what we want is actually put it back in the memory case. So someone can recall the situation, but like consciously instead of just having like flashbacks. And so what seems to work is behavioral and cognitive therapies, where the person actually like rewrites the event again and again and again, just to try to like rebalance the balance between hippocampus and amygdala, and try to like decrease a bit the amygdala, the activity in the amygdala, and give the hippocampus all the room it deserves. And it's something that seems to work way much more than the like drugs therapies.
Okay, great. Thank you very much. And the final question is for Dr. Maina. And to end on a positive, hopefully positive note, I would like to know what is your, how you use your work to give hope to future generations.
I think today we are living in the climate change area for sure, but we are also living in the technology era. We have a powerful technology. So the model that we are running basically today, I will need like 200 computers to be able to run just one day of what's happening on Earth. So I think that this technology we know for sure is going to change. So for the future generation is be able to combine the technology that we are developing to use it very well, not as social media or whatever things that we are going to do to fight for climate change. So that increase in the computing power gives me hope today.
Great, thank you very much. I would like to thank all of you today. Thank you very much for those 3 outstanding presentations and your insight. I'd like to also thank the Office for Science and Technology for partnering with DEFI today. And thank you everyone for being here with us today and engaged in this wonderful conversation. So thank you very much and take care.
Thank you.
Take care.